Now we will discuss what is the sequence of events that actually happens for a foodborne disease. And a sequence would, of course, would be there in every kind of food infection. Every food infection would follow a certain sequence. And the understanding of this sequence is very important because once we know this sequence, we can know the source of pathogens, the way the pathogen is transmitted to food, the source from where it comes, the way it spreads, the way it grows inside our foods, and all different routes that can lead to this end result, that is disease or foodborne infections. So once we know this in detail, it would be possible actually to break this sequence. And breaking of this sequence is very important because by then we can reduce the disease burden or happening of foodborne disease. So that is why it is important to understand what is the sequence of a foodborne infection for a particular pathogen, and that would give us information how we can break this sequence. We can avoid this happening of undesirable outcome, which is foodborne disease. So if we talk in terms of sequence or steps that are involved, the very first step, that is, there has to be a source of pathogen. That means there must be some source from which a pathogen comes or gets into the scene. And then from that source, the second step is it should contaminate your food. As long as it remains within the source and it does not contaminate, there is no concern. But once this is contaminated, this has contaminated your food, then of course now we are in a trouble. And a trouble begins in the third step, especially when we consume that food that has been contaminated with a pathogen. That pathogen may be a virus, or maybe a bacteria, or maybe any other parasite. So it means now we have consumed a food that has got this pathogen, or that has got this microorganism or any other parasite. So that means we are now exposed to this pathogen. So once the fourth step is obviously it would lead to infection, whether it's a virus or a parasite. The infection is the last stage. If we talk about these bacterial pathogens, and especially the toxicogenic mold, by toxicogenic mold I mean the molds that grow on your food and they release the toxic compounds inside your food, and those toxic compounds are basically responsible for the disease that can happen due to this food, due to the fact that it comes from the food. So that's why it would be called as foodborne disease. And we need to have certain conditions under which these molds or bacteria would be growing inside the food, and these bacteria or molds or other parasite would need to have conditions that actually support their growth within the food. So if we can stop these kind of conditions or make the condition unfavorable for the survival of these pathogens within food, that would be a healthy outcome for us. But if this is not the case, if the food supports the growth of these parasites or pathogens, then of course the foodborne disease would be obvious from this fact. But there might be cases in which this thing is not important. For example, the survival or growth of these parasites are not that important. For example, this E. coli, especially this very serious strain of 157H7, this is a serious foodborne issue basically. So once this is carried by food, even the food does not sustain the growth of this virus. That is not serious here, because even if it's there in your food for a very smaller concentration, that may lead to the foodborne infections in other people. So this is, maybe we can say this is an exemption where only this E. coli needs to be present in your food to create infections. Even the conditions do not favor the outgrowth of this pathogen. And then the intoxication of food from food would happen if there is a sufficient level of toxins that are present, because the toxin would be dependent upon it is dose dependent. If we have higher dose, we would have higher impact of these doses. In case of bacteria, we are concerned at how many number of viable microorganisms we are actually consuming. And these viable microorganisms, once they are consumed, then it would be dependent upon whether they can survive in our stomach or digestive system or not. If they can survive, that means that it is a serious concern. If we talk about these toxico infections, the viable microorganism that we consume through food, there could be two scenarios. For example, one in which the microorganisms are consumed in a smaller amount, but they can actually grow inside our stomach. For example, this cholera pathogen, Vibrio cholera. So once this can grow inside our stomach in the stomach conditions, then even small amount of this pathogen would be serious, because the pathogen can grow and then it can create disease. But in other cases, maybe there are other bacteria or pathogens that cannot grow inside your stomach, so we need to have higher number of these pathogens consumed as a live cell, and then they can create serious effects. So it would depend upon the survival of bacteria within our systems. If they can survive, we need smaller amount to get sick, and if they cannot survive, we need higher amounts of the pathogens. So summarizing all this, we need to have a reservoir of pathogen from where the pathogens get into your food. It would contaminate your food. So this food, contaminated food, it might be containing this viral or parasitic infection where the growth of this virus or parasite is not required. So this is one scenario. The other scenario is that the growth of pathogenic bacteria or molds, it takes place within the food during storage. And that leads to three scenarios. One is intoxication, where the consumption of food containing toxins would lead to the disease. Another scenario is it does not create any toxins, but it creates infection. So this infection is basically due to the consumption of food containing live cells. The third condition is, as we discussed, is toxico-infection, where the live cells are consumed and they form toxins inside the host. In other words, this is the summary of the sequence of events that take place in case of microorganisms for creating any foodborne disease.